Om Swastiastu, Assalamualaikum warahmatullahi wabarakatuh, Namo Buddhaya, Shalom, and greetings to everyone. First of all, us from Small Group Discussion B5 would like to thank all parties involved for having given us the opportunity to present our paper on blood and non-blood sucking flies. And here's a brief introduction of all our members, with myself being Justin Kin Adinata Santosa, with the last three digits of my student number being 106. And that'll be all from me for now, and I'll pass it off to my friends. Thank you.
Greetings everyone, my name is Yana Siraldijit with the last three-digit number 119. Flies are infamous for being the factors of many diseases such as dysentery, diarrhea, typhoid, and cholera. These diseases may cause a high morbidity and mortality rate, especially in developing countries. In fact, typhoid fevers account for hundreds of thousands of deaths annually around the world. Flies are divided into blood-sucking and non-blood-sucking flies. Blood-sucking flies usually take in the infective microorganisms while consuming the blood of an infected host, and later transfer it into a new host while consuming their blood. Conversely, non-blood-sucking flies are capable of transmitting pathogens from a host to another without drinking the blood. The problem formulation of this research include what are the blood-sucking and non-blood-sucking flies, how do these flies spread disease, and what are the diseases spread by these flies. This paper has been written for the purpose of having a better understanding regarding blood and non-blood-sucking flies and their effects, especially diseases on humans. Theoretically, these research serve as a basis for understanding blood and non-blood-sucking flies and the diseases they bring, and also serving as a reference for future studies. While practically, it may increase knowledge and awareness in order for advances in healthcare and health standards to be made.
Okay, so first, my name is Fairoza Husnia Adiba with three last digit number 102. Next. Okay, so what is blood-sucking flies actually? So blood-sucking flies are the type of flies that feeds on blood, and by that they be able to transmit disease agents to the host body and can cause harm, which is like disturb our circulation system. So there are many kind of blood-sucking flies. For the first one, I'm going to talk about horsefly and deer fly. So they are from the family of Tabanidae, and the roles of the sucking blood is done by the female group, which is for oogenesis, and they leave deep wound cut to the skin because their scissor-like mouth part, and that may cause pain to the host. So for the morphology, the larva stage is spindle-shaped with whitish color, and the pupa stage is tan with eyes, legs, and wings that can be seen, and the adult stage is where we can differentiate between them. So you can see here in the table that morphological characters used to differentiate between them. So the first one was body length. For the horse flies is 10 to 30, and the deer flies is 6 to 11. And the antenna for the horsefly is short, and the flagellum is greatly enlarged. Meanwhile, the deer flies is not, and the antenna is long. So for the ocelli, for the horse flies is vesicle or lacking, and the deer flies it is present. And for the wings, for the horse flies it is clear, meanwhile the deer flies it is distinctly bent. And the apical spurs on hind tibiae was for the horse flies is lacking, and the deer flies is present. Okay, so next.
Hello, allow me to introduce myself. My name is Tracy Odela Jedalia Kema with last three-digit NIM number 115. The next type of flies are stable flies or Stomoxys calcitrans. Their size is around 6 millimeters with 4 stripes with a dark gray color located behind its head and thorax. They are usually very common in sunny locations, and their biting activities usually occur around the late afternoons or early mornings. These species usually develop in filthy or dirty organic substrates. The next are black fly or Similidae. Black flies are known as buffalo gnats. Their size is very small, which is around 3 millimeters in length, with large wings and a humped back. Their color are usually black, but some species with orange or yellow colors are found. They are usually found in flowing waters, such as rivers, and common during the early summers and late springs. Next are biting midges or Ceratopogonidae. Biting midges are also called no-see-ums because they are very small, around 1 until 2.5 millimeters. They are usually found along ocean shores, lakes, or rivers, and bite during the day or night. Their breeding locations are varied, but most of them are located in organic substrates. Next are sandflies or Psychodidae. Sandflies belong to the Phlebotominae subfamily in the Psychodidae family. Their size are no more than 5 millimeters and have slender, long legs with broad V-shaped wings and a brownish-gray scales. They usually target amphibians, reptiles, and mammals at night, and they usually live in moist decayed plants in nature or dark and humid places in domestic areas.
Introduce myself. My name is Kevin Nathanael and my last 3 digit is 105. Right now I'm going to explain the life cycle of Glossina sp. First of all, the eggs will hatch in the uterus after 2 until 4 days and become first instar larva, and then transforming into the second instar larva for 1 until 2 days, to the third instar for 1.5 and 2 days, and then become fully developed until 2.5 days. After that, the larvae will molt and become pupa, and then after the next 30 until 40 days, the pupa will break and hatch into an adult. Next, I'm going to explain the life cycle of chrysops. First of all, the eggs will hatch into the larva after 2 until 3 days. The larva will keep growing until 6 and until 13 weeks, and then become pupa, and then complete it after 1 until 3 weeks and become adult. Nithabanus sp. life cycle is similar to the chrysops, so I will skip that. And right now I'm going to explain about the life cycle of Phlebotomus. First of all, the eggs will hatch after 20 days and become larva. The larva will undergo 4 stage of development for about 30 days and then become pupa, and after 6 to 13 days it will become adult.
Let me introduce myself. My name is Samantha Petrona Theobald with NIM 22025111108. Flies are a type of ectoparasites which play a role in becoming a vector of disease caused by microorganisms and parasites. Flies are divided into non-blood-sucking flies and blood-sucking flies. Non-blood-sucking flies are a type of flies which do not suck or drink blood, and become a vector of several bacterial types of transmission by spreading through the food that has been eaten by flies. Morphologically, Musca species fly have a proboscis mouth with a widened tip and sponge-like structure to absorb food. Forward wing venation which form a slightly oblique angle close distally. Brown thoracic side, 4 stripes, and yellow belly. For the characteristic, Musca species mostly attract to hips and animal feed. Considered omnivores, able to reproduce in dirt, wet fodder, and other organic ingredients, often associated with hygiene problems and highest domination rate due to high adaptation capability, reproduction rate, and mutability rate. For the habitats, Musca domestica common in livestock environment and any other place or cosmopolitan.
Let me introduce myself. My name is Ida Bagus Tresna Indra Putra Pramana with the last 3-digit number 104. Continuing my friend's explanation, the next type of non-blood-sucking fly is Fannia sp. So morphologically, Fannia sp flies possess licking-type proboscis and parallel fourth-wing venation with a general shape from medium to smaller from Musca domestica, with black body and legs. It could also be found in livestock closures if it is located near poultry house. Fannias are infamously known for their ability to exploit decomposed material for their reproduction purposes. They can also be found in numerous stages of decomposition. Fannia sp breeds in humans and animals' feces and also in rotting plants' parts. Presence of Fannia sp often found in poultry facilities and often attacking laying hens. So the next type of non-blood-sucking fly is Lucilia sp. So morphologically, Lucilia sp flies possess a proboscis equipped by labellum for absorbing and leaking fluids function. The fourth wing venation is angled sharply and slightly curved inward. Lucilia sp have a varied body color such as metallic green, metallic blue, and brownish green metallic. These types of flies also possess a round to oval body form with a variety of length. The gender of Lucilia sp is also distinguished by its eye structure. The female possesses a separate left and right eye, while the males are merged. Lucilia sp lives around the watercourse, heaps of garbage, and animal waste, and also stingy pungent.
is Natasha Pramatia Putri with student number 2202511111. I will explain about the transmission and ingestion of blood meal flies. Blood meal flies can be the vector agents for transmitting disease. The general mechanism is the blood meal flies bite the protozoa-infected host such as human, and the blood meal flies will be infected by the protozoa through the blood of the protozoa-infected host. The protozoa grow in the body of the blood meal flies until infectious stage. The flies take a meal from non-infected host and transmit the protozoa. One of the examples is trypanosomiasis. The vector agent is sasa flies. The mechanism: sasa flies take a meal from the infected host, and sasa flies become infected by the protozoa. Sasa flies transmit the protozoa to the non-infected host by take a blood meal from it. The protozoa move to the lymphatic system and blood circulation of the host. Next examples is tularemia. The vector is deer flies. The mechanism: deer flies take a meal from the infected host. Deer flies become infected by Francisella tularensis. Deer flies transmit the protozoa to non-infected host by take a blood meal from it. And finally, the non-infected host become infected. Last example is leishmaniasis. The vector is sandflies, and then the mechanism: the sandflies take a meal from the infected host. Sandflies become infected by leishmania, and then the leishmania grow inside the fly until become infectious metacyclic chromatigogus, and then the sandflies transmit the protozoa to non-infected host by take a blood meal from it. The non-infected host become infected.
Okay, thank you for the opportunity. My name is Valentino Chandra Wijaya, which CC of new member is 116, and now I will explain the disease caused by blood-sucking flies in human. First is tularemia or rabbit fever. It's a disease caused by infection from lymphatic Coccobacillus Francisella tularensis, which is transmitted by bite of deer flies. Tularemia consists several types, namely ulceroglandular tularemia, glandular tularemia, oropharyngeal tularemia, and etc. Common symptoms of tularemia are as follows: fever, noise, vomit, rash, and others, and several special symptoms depending on the types. Treatment for tularemia is used streptomycin, gentamicin, doxycycline, and others. Prevention can be done in several ways, such as keeping close flame and using insect repellent on exposed skin and clothes. Next. Leishmaniasis, which is a disease caused by infection Leishmania protozoa, which is transmitted by flies. Symptoms and treatment are distinguished based on the three forms. Cutaneous villus lump can be treated with the cryotherapy and anti-leishmanial drugs. Mucosal, skin sores, and tissue damage can be treated with the drugs liposome, miltefosine, and amphotericin B. Visceral will be caused fever with low short-term enlargement, and anemia can be treated with the liposome, amphotericin B. And the prevention can be done in the several ways, such as using insect repellent on exposed body part and clothes, and using bed nets and insect spray. Next. African trypanosomiasis, or also known as sleeping sickness, is a disease that mostly occurs in Africa, caused by two protozoa, which are Trypanosoma cruzi capiense and Trypanosoma cruzi rudisiense, which is transmitted by a bite from tsetse flies. This protozoa will cause different effects depending on which tissue or organ is infected, like in liver, heart, and brain. The symptoms that occur in skin: there will be bumps in the lymph nodes and blood. There will be a rash and enlarged lymph nodes, and in brain and cerebrospinal fluid, there will be drowsiness, loss of talent and concentration, and sudden fall asleep. The treatment of this disease use crimamine, pentamidine, melasoprol, and nivolumab. And the prevention can be done in several ways, such as avoiding things that the habitat of tsetse flies, wear clothing that cover our skin with the neutral colors, and using insect repellent. Thank you.
Hello, let me introduce myself. My name is Gabriela Fembers Davita with last 3 digits student number 118. The life cycle of Musca and Fania depends on their environmental circumstances. The life cycle begins with the egg stage, which is generally laid in the garbage, meet the human feces by the adult of the previous generation. The eggs are characteristically wide with a size of approximately 1 mm. When entering the larval stage, they will be faced with 3 instar phases consisting of larva 1, 2, and 3, which can be identified by their color, which in phases 1 1 and 2, the larvae are white and will turn yellow when they reach phase 3. After reaching the larva stage, houseflies also experience changes in their body anatomy with the appearance of spiracles as their breathing apparatus. They will also move to a dry area to enter the pupal stage. The larva skin will begin to shrink and form a puparium. The spiracles remain on the outside of the pupa as species determinant. They finally reach the young fly's phase and will undergo a resting phase, which at the same time their wings will begin to develop and experience a change in color to black. On the other hand, Lucilia sericata tend to lay their eggs on a fresh carcass. In the larval stage, Lucilia sericata also experiences the same instar stage as Musca and Fania. These larvae will later begin to migrate to dry areas to continue their life process. They become pupa, which will last for 7 days to become an adult. Adult flies that have emerged from the pupa will be metallic green, which has been equipped with a pair of antennae, eyes, thorax, and abdomen. That's all from me. Thank you.
Hi, my name is Neta with the last 3 digits of name 112. Amoebiasis, diarrhea, and typhoid fever have the same transmission factor, namely Musca domestica or housefly. In addition, these 3 diseases spread through fecal-oral contamination, contaminated food and water, poor hygiene, and hand contamination from contaminated surfaces.
Hello, my name is Bernard Karel Kuresmafati with the last 3 digits of my student number is 103. So now I'll present to you topic 2.8, disease caused by non-blood-sucking flies in human and its prevention. The first disease is amoebiasis, or also known as amoebic dysentery, is an enteral parasitic infection that is common or often found in cases. This disease is caused by a type of amoeba from the Entamoeba group. The prevention of this disease is, the first, do not defecate indiscriminately. The second is washing hands with soap. The third is managing drinking water and household food. The fourth is managing household waste. The fifth is ensuring the safety of liquid waste disposal channels. The second disease is diarrhea. Diarrhea is a condition in which patient or a person experience more than 3 liquids or watery bowel movement in 24 hours, with or without mucus and blood. The prevention of this disease, the first, access to safe drinking water, use improved sanitation. Third is hand washing with soap, exclusive breastfeeding for the first 6 months of life, good personal and food hygiene, health education about how infections spread, and the last one is rotavirus vaccination. The third disease is typhoid fever. Typhoid fever is an acute gram-negative bacterial infection disease caused by Salmonella enterica serotype TP and paratype. The bacteria enter through food or drink that is contaminated with the feces of sick people or carriers. The prevention of typhoid fever can be done by administering vaccine. The first vaccine is T21 vaccine with efficacy of 68 until 82%, and the second vaccine is Phi capsule polysaccharide vaccine with efficacy of 70 until 80%. These 3 diseases are usually caused by consuming food or drink that has been contaminated with the feces of sick people or carrier that carried by houseflies or Musca domestica. That's all from me. Thank you.
So, in conclusion, flies are insects whose bodies are divided into 3 separate segments and has 1 pair of wings, thus classifying them into the order Diptera. They are known to spread diseases by being vectors. From a medical point of view, flies can be either blood or non-blood sucking. Blood-sucking flies spread disease by transferring contaminated blood from one host to another, while non-blood-sucking flies spread disease by transmitting viruses, protozoa, bacteria, etc. Some notable blood-sucking flies include deerfly, horsefly, stablefly, blackfly, biting midges, and sandflies. They are known to be the causes of the diseases trypanosomiasis, tularemia, and leishmaniasis. Non-blood-sucking flies, such as the Musca species, Fania species, and Lucilia species, are known to spread amoebiasis, diarrhea, and typhoid fever. And on that note, that concludes today's presentation. We sincerely appreciate your time and attention. We do apologize for any mistakes that we have made. Should you have any inquiries, please feel free to contact us by the email address listed down below. And finally, allow me to close it off with Parama Santi. Om Santi 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 Om. Wassalamualaikum warahmatullahi wabarakatuh. Shalom, Namo Buddhaya, and have